The overall goal of the following experiment is to measure the activity of intracellularly expressed sodium proton exchangers. This is achieved by first selecting cells that express the intracellular sodium proton exchangers at the plasma membrane by proton-killing selection. As a second step, the selected cells are acidified, and then the intracellular pH recovery is measured to confirm the sodium proton exchange activity of the cells. The fast kinetics of the ion transport are then measured to determine the affinity constants for the transported cations and inhibitors. Ultimately, monitoring the changes in the intracellular pH and fast kinetics of the selected somatic cells allows the functional characterization of the intracellular protein exchange.
The main advantage of this technique over existing methods like expressing mutant transporters or using permeabilized cells is that it enables the expression of wild-type intracellular transporters at the plasma membrane, making possible to directly measure their function. Demonstrating the procedure will be Dr. Mallorie Poët, a research scientist from my laboratory.
To begin the proton-killing procedure, first, incubate the cells expressing the intracellular sodium proton exchanger of interest in loading solution at 37 degrees Celsius in a carbon dioxide-free incubator for 1 hour. Next, aspirate the loading solution and wash the cells twice with fresh medium. After the second wash, aspirate the rinse medium and incubate the cells in recovery solution.